Hello everyone, this is Tracy Koch again, and I'm going to be lecturing today on Chapter 12, The Genetics of Cancer. All right, so cancer is, as we all know, a disease of unregulated cell growth, and it serves no useful purpose. So the first slide, which statement is true? All cancer is genetic, all cancer is inherited. I think this is really interesting because I always sort of think of some cancers as being genetic and some cancers not. But the fact of the matter is all cancer is genetic because it starts with cellular DNA abnormalities. So it is indeed genetic. All cancer is inherited. Well, that's a little more complicated, and we'll talk about that. Cancer facts. All cancer is genetic in that it is always caused by changes in the expression of genes involved in cell division. So that's what I was referring to earlier. It has to do with cellular abnormalities in the genetic material. Cancer cells transmit gene changes from one cell generation to another. So as cells are dividing and going through meiosis, the genetic abnormality is transferred from one cell generation to another. But cancer is not inherited from one human generation to another. So these are some important concepts for you to remember when we're thinking about the genetics of cancer. Specific mutations in genes regulating cell division that occur in germline cells can be inherited from one human generation to another and greatly increase the risk for cancer development. So, cancer is a disease of the cells. That's an important fact to remember. And you can have that germline abnormality inherited from one human generation to the next. Cancer arises from normal cells that have been altered at the DNA level. The most common mechanism of malignant transformation involves suppressor genes. So your suppressor genes are mutated, and that leads to unregulated cellular activity. So we might inherit the risk of cancer development from our parents, but that is not the case with most cancers, as we'll talk about here a little more. So I would say the key thing to remember with this particular concept is that cancer is indeed a disease of the cells and not just an organ disease. And the cells lose function and change appearance, so that's why they're abnormal. Okay, let's move on. Even one cancer cell can generate widespread metastatic disease, as we know, and that's referred to as the monoclonal origin of cancer. So three things need to interact in order to develop cancer. You need to have an environmental exposure to carcinogens, the health of the immune function, and a genetic predisposition. Normal cells. It's good to review this because then it kind of makes sense when we talk about cell abnormalities with cancer development. So normally our cells are very orderly, well-regulated, their functions are differentiated, and they follow a specific morphology. They have a small nuclear to cytoplasmic ratio. They respond to signals for apoptosis, which is cellular death. That's programmed already. They're euploid, which they have the normal number of chromosomes for our species, and they're tightly adherent. So that's a normal cell function. Early embryonic cells, they're rapid, continuous cell redivision. They re-enter the cell cycle immediately after mitosis. Anaplastic morphology, they're small, round, Anaplastic means that they rapidly divide. And they have a larger nuclear to cytoplasmic ratio. They are loosely adherent, so they don't have the cell adhesion molecules present, and they're able to migrate. And they're euploid, which is the only feature in common with mature differentiated normal cells. So a benign tumor, the cells of a benign tumor would have specific morphology. They're non-migratory. They're usually euploid, so they have the normal chromosomes. They're small, or the small nuclear to cytoplasmic ratio. Their functions are differentiated. They're tightly adherent. They grow by expansion. They don't invade. So they do have inappropriate growth, but they don't invade like metastatic cells, which we'll talk about in a minute. Cancer cells. Okay, so here are the differences with cancer cells. They are rapidly, continuously dividing. So they re-enter the cell cycle immediately after mitosis. So if we're thinking back a couple slides ago, that was also a feature of early embryonic cells. They don't respond to apoptic signals. So they don't respond to those signals that cause spontaneous or programmed, rather, cell death. So these cells have lost that ability to destroy themselves, for lack of a better term. The genetic regulation of cell division is lost. So there is no regulator. I think about in the old I Love Lucy show where the candies are coming down the conveyor belt really fast. So that regulation is lost in cell division when you're looking at a cancer cell. They have anaplastic morphology, so they're all small and round. They have the larger nuclear to cytoplasmic ratio, again like the early embryonic cells. They have no differentiated functions. They are loosely adherent, so they also don't have any cell adhesion molecules. So this, when we talk about metastasis, we'll come back to this fact that they're loosely adherent. They are able to migrate, and they are aneuploid usually. Okay, next slide. Anaplastic morphology is they progressively lose their parent cell features. They become small and round, and they all look alike, even among different cancer types. They have the small nuclear to cytoplasmic ratio. So here is an actual slide or photograph of cancer cells. And they have no fibronectin, so they can kind of pile up on each other, but they're still loosely adherent. So it means it's really easy for them to break off and migrate elsewhere through the circulatory system. So if the cells just stayed in one area, like with, let's say for breast cancer, if it just remained in the breast, it probably wouldn't cause death because the breast is not critical tissue. It's the fact that the cancer metastasizes, it's the metastatic disease that, makes it fatal. When the cancer cells metastasize to a vital or a critical organ, they disrupt this function of the organs and tissues, and that's what leads to death. Other cancer cell features, they lose the parent cell differentiated functions. They stop producing cell adhesion molecules, so they gain this lytic enzyme on the cell surfaces, and the tumor breakage and release of cells into the bloodstream. So the cell membrane is very open, and it doesn't need insulin to metabolize glucose. So the glucose just moves in to the cells, and so this again kind of speeds up that whole process of cellular growth and mitosis, and you can see these cells just grow wild. Tumor cell migration and invasion into other tissues, which is metastases. And this is the metabolic advantages that I was just referring to about the glucose. It allows the cells to continue to grow even when the host nutrition is poor. And this is all related to genetic alterations, or alterations in the genes. So this is a picture of abnormal chromosomes. Abnormal chromosomes, or the number, either in number or in structure, is aneuploidy, aneuploidy. That's aneuploid, is the abnormal chromosome number or structure in a cell. That's what makes it aneuploid. As you increase malignancy, the more abnormal that those cell chromosomes are. However, some chromosome rearrangements are helpful for us. They help us diagnose and predict prognosis in certain kinds of cancers. So with chronic myelogenous leukemia, CML, in 90% of the cases, they have what's called a Philadelphia chromosome. It's a broken number 22, excuse me. And this broken number 22 is what makes it easier to diagnose. But we've also learned that the ones that have the broken number 22, or the Philadelphia chromosome, is more responsive to therapies. So that's a helpful thing to know as well. Malignant transformation. So we know about these cancer cells, that they're these rapidly dividing. They don't follow any of the rules. They don't adhere to one another. They migrate. They are just these rogue, crazy cells. So let's talk a little bit about how this happens. All right, so the process of changing a cell from a normal phenotype to a malignant phenotype is the malignant transformation. It occurs in a stepwise, time-dependent, well-characterized progression, so it follows a pattern, and it involves, again, cell alterations at the gene level. So here's a little diagram of the mutational event, the latency period. So you have your normal cell here, the mutational event, the latency period, and you have this continued exposure to promoters, which we'll talk a little more about that, and that's what leads to the development of the cancer cells. This diagram kind of goes through the different steps: initiation and promotion, and the continued promotion that take the normal cell into a cancer cell, and then allowing it to become more and more malignant, and then to a metastatic state. Geez, I'm really tripping on my words today. And that's what causes metastatic disease. This is a progression over time. So the example Workman uses in the book, that you don't usually develop lung cancer a week after you smoke your first cigarette. It happens over time. All right, so first step is initiation. This is the damaging of the DNA, either directly or indirectly, by agents or conditions that result in the permanent alteration of gene expression. It doesn't interfere with the altered cell's ability to reproduce. So some of the initiating agents are chemicals, radiation, which is a physical agent, or biological, like a virus, or inherited DNA mutations. So the initiation results in permanent gene mutations or changes, so this is not reversible.
Well,
Mutations in the genes regulating cell growth lead to cancer development. So what happens is you have these rapidly dividing cells. There's no shutoff mechanism. They don't die like a normal cell. They don't have the normal cell life cycle, and that's what develops into a malignant cell. Oncogenes are the abnormal gene that predisposes cells to develop into cancers. So they're altered in such a way that they're stuck in a state of constant activity. So this drives that uncontrolled growth that underlies tumors. The suppressor gene products are the driver and the brake, like in the car analogy. So the suppressor genes will slow down or shut off that process. Suppressor gene products regulate cell growth. It limits and inhibits the expression of the oncogenes, and it does induce apoptosis when it's needed. So here are some examples of suppressor genes, and you likely recognize BRCA1 and 2. So you have your suppressor genes out here. You have your overexpression of your oncogene, and the suppressor genes are either lost or mutated in a cancer cell, and that's why you have this cell growth and proliferation that leads to a possible cancer. Okay, so suppressor genes and associated cancers. Here are some a little chart on that, and that's in your textbook. Oncogenes are normal genes that regulate the cell cycle. The code for the proteins relay signals to the nucleus. The signaling process begins at your cell membrane, and it relays the signals through the rest of the cytoplasm. It ends in the nucleus with the activation of transcription factors that help move through the growth cell cycle. Examples of pro-mitotic oncogene products are various cyclins, aurora kinase, and various cyclin-dependent kinases. These are examples of oncogene mutations and associated cancers. The main defect in carcinogenesis is inappropriate and excessive oncogene expression, regardless of the specific cause. Back to cancer facts, mutation in your suppressor genes. So even one allele that is mutated reduces the ability of the suppressor gene products to inhibit the activity of the pro-mitotic oncogenes. So even one minor defect in the suppressor gene can cause the abnormal cell division, and then the cell cycle takes off with a the cell cycle takes off and you get the rapidly dividing cells, etc., like we talked about earlier with cancer development. Mutations in the suppressor gene alleles or the DNA repair genes cause most cancer development. Mutations in oncogenes, although occurring less frequently, result in loss of oncogene sensitivity to your suppressor gene regulation, and both types of these mutations lead to oncogene overexpression. Here are some examples of the DNA repair gene mutations and associated cancers. And here's a little graph of the car analogy where the car has both sets of brakes. The suppressor gene alleles are working properly in figure A, so he stops in plenty of time before he hits the trees. In figure B, the front brakes or a suppressor gene allele are not functioning, and the car takes a longer distance to stop, but it still doesn't hit the trees. And in the diagram C, the front and rear brakes, so both suppressor genes are non-functional, and the car cannot stop, and it hits the trees. So that's kind of a long explanation about initiation. But basically, initiation in cancer development is the mutating cell DNA is either suppressor genes can't perform the regulating cell growth, or the oncogenes become resistant to suppressor gene control. So that really is what initiation kind of boils down to. So it's either the mutating cell DNA, the suppressor genes cannot perform their regulating cell growth, or the oncogenes are resistant to suppressor gene control. All right, so let's move on to promotion. Promotion is the enhancement of the expression of the gene that's altered during initiation. Promotion usually increases the growth rate of the cells and is a reversible event. Promoters can be external to the cell or internal, so enzymes, hormones, natural growth factors. If a promoter isn't strong enough, or exposure isn't often enough, the cancer may never develop, even though you've had the initiation step. So this again, we're back to our diagram where we have our normal cell, the mutational event, and then the exposure to the promoters leading to the cancer cell. And then again, our continuum where it shows the initiation, and you have the promoters that lead, that exposure leads to the metastases. Progression is the continuing change over time of a cancer cell population, causing them to be more malignant or more aggressive, less normal in appearance, function, and growth regulation. So you have increased growth, increased invasiveness, biochemical changes, morphological changes, and metastases. Less malignant cells more closely resemble our normal cells from which they arose. But as cancer cells become less normal, they become more malignant. They're poorly differentiated. So when you look at a biopsy report, if you see highly differentiated or well differentiated, those might be less malignant. But as you see poorly differentiated and undifferentiated, those are more malignant cells. All right, so we are on metastases. Metastases is the special ability of the cancer cells to invade other tissues. Remember we talked about benign tumors, how they grow by expansion and they don't really invade? Well, metastatic tumors do invade. They have a special ability to either form secondary tumors or by direct extension separate from the original tumor, go through the circulation and invade other areas. Okay, sporadic cancer is, it occurs as a result of an environmental exposure or unknown factors. So there's no real pattern of inheritance within a kindred. The mutations through carcinogenesis disrupt the regulation of cell division, and it's not present in higher than expected levels within 3 family generations. So when you draw a pedigree, you don't see a more common or a more observable pattern, and you don't see higher than expected cases in a 3-generational pedigree. Sporadic breast cancer occurs without any observable pattern of inheritance. It's not present in a family at a higher than expected level, and affected individuals are all postmenopausal women. And here is a pedigree demonstrating that. Familial cancer is a cancer that occurs at a higher than expected rate frequency within a kindred. It doesn't have any observable pattern of inheritance, but family may have a higher than expected incidence of several cancer types. And those who develop cancer are typically older in a familial cancer, usually over the age of 60. Familial breast cancer, again, occurs without an observable pattern of inheritance, present in a family at higher than expected levels, so greater than 1 out of 8, and the family may also have higher incidence of other cancer types. Affected individuals are all postmenopausal women. And here's a pedigree that demonstrates that. I believe these are all in the textbook too. Hereditary or inherited cancers, they occur within an observable autosomal dominant pattern within a kindred. So you would do a 3-generational pedigree and see a autosomal dominant pattern. Diagnosis are at much younger than expected ages, and this is caused by the germline mutations in the suppressor genes. Remember I mentioned that earlier in the lecture, and this is where that comes into play. The risk is inherited, but the actual cancer is not. The risk is greatly increased, but it is not an absolute that you will develop the cancer if you're in that kindred. Hereditary breast cancer, observable autosomal dominant pattern of inheritance. It's present in a family at much higher than expected levels, often associated with other cancer types, and the affected individuals are women and men of any age, although we see it more than in women, and it's typically it is bilateral disease. And here is a picture or a pedigree showing that. So, kind of recapping this section of the lecture, some key elements. Inherited cancers tend to appear at younger ages. The sporadic cancers tend to be in older postmenopausal women when we're talking about breast cancers. Okay, so there are a few case studies, and these are not the ones in the textbook, I don't think. So here is a pedigree. So this pedigree shows Belinda, Bonnie, and Brett. And Belinda is 56. She has 2 daughters and a son. She has just been diagnosed with a recurrence of lobular breast cancer, treated by a lumpectomy and radiation 4 years ago. Her father is still living at age 82. Her mother, Anne, died of ovarian cancer at age 36, and she was diagnosed at age 34. Her sister Bonnie was diagnosed with breast cancer 6 years ago at age 32 and died 2 years after her diagnosis. Oh, yeah, sorry, this is, she died 2 years after her diagnosis. She was married and has no children. Her brother Brett, now age 45, underwent surgery and chemotherapy last year for breast cancer also. He has 1 son, 1 daughter who are teenagers. Her mother had 1 sister, Amy, who died of abdominal cancer when she was 34, and 1 brother, Alan, who died of bone cancer at 41. Her father's twin brothers died at Bergen-Belsen during World War II, and Belinda's daughter, Connie, is 30 and recently diagnosed with breast cancer. She has 2 sons, ages 1 and 3. Her older daughter, Catherine, is 32. Son, Carl, is 26. Neither Catherine nor Carl have any evidence of disease. So I think in my, Connie is actually Caroline, who is 30 and recently diagnosed with breast cancer. Anyway, so this is their pedigree. So the pattern of inheritance is autosomal dominant. Males and females are affected in nearly the same proportions. The affected individuals have affected children, and the transmission is from parent to child. So genetic testing for these folks might be beneficial, just because if they're positive, they could still, develop ovarian cancer, and Belinda might have children who have inherited the mutated allele. And if Belinda is positive for the mutation, each of her children has a 50% chance of inheriting that mutation, and they could develop breast or ovarian cancer. So would be nice to pursue genetic testing for this family so that they knew what they were dealing with. So here's another case study. With showing a pedigree with another cancer, and I don't think this one is in your textbook. But Martin is 68, and he's indicated down here in the corner. He was diagnosed with colon cancer. His sister at age 65 was also diagnosed with colon cancer last year. They have a maternal aunt age 90 and 1 paternal aunt age 75 with breast cancer. A maternal uncle, age 72, had colorectal cancer, and 1 paternal uncle who had prostate cancer. A maternal great-aunt died of childhood leukemia, and a second maternal great-aunt died of breast cancer. The maternal great uncle died of lung cancer at age 64. So here's their pedigree. And there's no obvious transmission pattern depicted in this. So although there's no clear transmission pattern, the number of people in this kindred with cancer is greater than what would be expected by chance alone. So this would best be defined or identified as a familial cancer. And really, there wouldn't be any genetic testing because there's no known mutation that increases the risk for the cancer development that we see here in this pedigree. So that wraps up our lecture on chapter 12, the genetics of cancer. And there is a case study in the textbook that is assigned for you to do. Thank you.